Hey guys, we're going to finish up this chapter by talking about cellular responses. So this is what is it that happens after this signal has been received and sent through the cell. So one option is regulation within the cytoplasm. So you can have rearrangement of the cytoskeleton, you can have ion channels that are going to open or close, or you can have a change in the cell's metabolism. You could also have regulation of transcription. And so this would be a case where activating protein synthesis for a new enzyme, so you're turning on a gene to create new enzymes, or you can have transcription control factors that are going to be activated by protein kinases. So what we're looking at here is, again, we have our G protein link system. We're activating relay proteins, we're activating these protein kinases, and eventually that protein kinase is going to activate a transcription factor. This transcription factor is going to transcribe a gene into mRNA. The mRNA transcript will then leave the nucleus, go back out to the cytoplasm so that a new protein can be made. So a question that I want to kind of take a look at is, if you're looking at two different kinds of cells, so let's say like a liver and a heart cell, and both are exposed to ligands or to these signal molecules, one might respond and the other might not. And so why is that? And one really simple reason could just be that different cells have different receptors, but we also have some alternative explanations as well. And so here we have cell A, and so the signal molecule is attaching to the receptor. You're activating these relay molecules and you get a certain cellular response. In a different cell, though, you could have, again, the same signal molecule, the receptor protein, but instead of just having the same relay, here you could alternate two different branches. So you have two different relay proteins activated. So here you're actually going to get two different responses. So response 2 and 3. In cell C, what we're looking at here is we've got the same signal molecule, but here we have another signal molecule coming in and binding to a different receptor. And so we have the same relay protein, but here this one is also initiating a relay protein, and what's happening is that you have crosstalk. So essentially one signal molecule is affecting how the other one is expressed, and so you're getting a different response. And now cell D, again, is a little bit different. So in this case, the receptor is going to be different from the ones in the previous cells, and so you're going to get a different response. So let's take a look at signaling efficiency. And this can actually be increased by the use of something called scaffolding proteins. And these are proteins that hold groups of signal pathway proteins together. So it holds them or groups them together so that they can actually be carried to the receptor together. So here again we have our signal molecule coming in, a receptor protein, and you can see we have our scaffolding protein, which is going to bind to that receptor, and it's carrying with it three different protein kinases that can actually start three different pathways. Next, we're going to look at apoptosis. Apoptosis is programmed cell death. And so this is something that uses signaling pathways quite a bit. What happens is the DNA is going to get chopped up. Cells will shrink up and become lobed. And then pieces of the cells are going to be digested by specialized scavenger cells. So again, this is what happens when cells need to die. What you're looking at here is essentially how this happens with interdigital tissue. So we have like the start of a hand, and you can see it's currently webbed. Over time, that webbing is going to break down, but those are live cells, and so essentially all of these cells are receiving that signal that they don't need to be there anymore, and so apoptosis is taking place, and now you can see that we have the much clearer digits. This is just showing a white blood cell before and after that signal has been received. And so this is going to be a balance between signals for live or die. It can be triggered by damage to mitochondria. Cells can be received from neighboring, or signals can be received from neighboring cells, or you can actually have internal signals within the cell that trigger, this programmed cell death. It's involved in several different diseases, so things like Parkinson's, Alzheimer's, cancer. I'll show a link. This is just another example. Again, here you've got the death signaling molecule. It's going to meet with the membrane receptor protein, and it is going to activate this cascade right here, and it's showing that you've got the lobes forming on the cell as it starts to break down. So the summary for this chapter: don't get bogged down in the details. So you do need to know the 3 stages of cell signaling, and you need to pick one receptor, and you need to know in detail how it works. So pick one, pick your favorite, and get to know it really well. You need to know about the protein kinases, how they are activated. You need to know about cascades and amplification. You should know one example of a secondary signal molecule. Again, get to know it well. And then finally, you need to know about programmed cell death. So you need to understand that, understand the link between apoptosis and several of the diseases that we mentioned. And lastly, your lecture question for the day. So what are 2 ways that cells could be affected by errors in apoptosis? So what would happen to a cell if it wasn't receiving those signals the way it should? All right, thanks guys.